आ गया सर का डिस्प्ले, आ रहा है ना सर का डिस्प्ले? अब वॉइस चेक कर लेते हैं। हेलो हेलो, शालिनी मैम बोलना। हेलो शालिनी मैम।
यस सर, यू आर ऑडिबल।
ऑडिबल। कैमरा ठीक है?
बिल्कुल ठीक है सर कैमरा।
ओके,
थोड़ा सा वो थोड़ा ब्राइट नहीं है।
ठीक है, अभी है?
हाँ नहीं, थोड़ा कम ब्राइटनेस है।
अभी?
अब ठीक है, विजिबल है।
विजिबल है?
हाँ जी,
ठीक है।
ठीक है सर।
ओके, मैं अभी सर का कर रहा हूँ चेक ना।
गुड मॉर्निंग सर।
मैं सर का डिस्प्ले चेक कर रहा हूँ अभी सॉरी कपूर सर का नहीं हुआ है कैमरा।
गुड मॉर्निंग तेजवीर सर।
कर दिया रे।
गुड मॉर्निंग।
चेक करूंगा, मैं उसको बोलता हूँ यानी म्यूट कर रहा हूँ, ठीक है? आ रहे हैं। ये दे दीजिए सर। शालिनी मैम आ रही है आवाज?
यस सर, ऑडिबल है।
कैमरा क्लैरिटी ओके?
कैमरा क्लैरिटी ओके है, थोड़ा सा पीछे धूप है ज्यादा। ओके है सर।
ठीक है। सर आवाज आ रही है? सभी ऑडिबल है सर?
यस सर, ऑडिबल है।
Shani, am I audible?
Yes sir, you are also audible.
Hello.
Yes sir, Kapoor sir. Good
Yes.
morning sir.
Good morning. Good morning.
Tej Vir sir, good morning.
शालिनी मैम स्टार्ट करें?
हाँ जी
गुड
सर।
मॉर्निंग टू रेस्पेक्टेड टीचर्स। गुड मॉर्निंग कपूर सर।
Good morning.
Okay. Welcome back, dear participants, to join with us in International Online Conference on Drug Discovery, Development and Lead Optimization. We will continue this session with our keynote speaker, Professor Dr. Tej Vir Singh. He is Professor Emeritus in Baddi University of Emerging Sciences and Technology and former faculty Department of Chemistry, Punjab University, Chandigarh. He is lifetime member of Chemical Research Society of India, IISc, Bangalore. Lifetime member of Punjab Academy of Sciences, Punjab University, Patiala. He has more than 42 publications in international or national journals, more than 4 books, 14 PhD students he has guided, and he is currently giving consultancy to Reckitt Benckiser India Limited, Gurgaon, Pharma Affiliates, Panchkula, Mill Specialities, Derabasi, and Chemical Resources, Panchkula. Now, may I please request Professor Dr. Tej Vir Singh, sir, to please share your presentation. Over to Tej Vir, sir.
माइक ऑन हाँ जी शाली मैम स्लाइड विजिबल है?
Yes, sir, it is visible.
Okay, at the outset, I would like to speak on synthesis, evaluation, and molecular docking studies of Galates as potent gastro-protective anti-inflammatory agents. Before I speak about it, I would like to acknowledge the help of my co-workers and co-authors of the research done on synthesis, evaluation, and molecular docking of Galates. The research workers were students such as Mamta, and the co-authors of the research papers were Professor Renu Chadha of UIPS, Punjab University, and Professor Maninder Karan of Punjab University, Chandigarh. Actually, we did this work on synthesis and evaluation of molecular docking, and then we published a paper on this one. First, I would like to show you the system, gastrointestinal system, by which you can have an idea that whatever we eat, it goes to stomach. And some problems of gastroenteritis can occur there only. What happens sometimes, some ulceration, stomach ulceration can occur because of the food or the acid generated in our body. Next. Here, I would like to show the structure formulae of some compounds like gallic acid. It has got 3 hydroxy group and 1 carboxylic acid group attached to benzene ring, though their substitution pattern has already been shown here. Now, if we convert the acid into ester, then certainly we will get the galates. But because of their solubility or less solubility in lipids, we converted the hydroxyl group into methoxy group. Therefore, we got trimethyl galates. Now, what happens that the action occurs with COX-1, COX-2, COX-3 with the galates. Now, we don't want to take the steroids, therefore, these are called non-steroidal drugs. Next. Yeah, there are some naturally occurring phenols containing 1 or 2 hydroxyl groups with benzene ring or with aliphatic site. The formulation let us say that we have 1A, 1B, 1C, up to 1I.
For the breakfast.
So these are all naturally occurring antioxidants of the derivative of 3-hydroxyphenol. As a matter of fact, the phenol moiety, or let us say the OH moiety, is essential for all such compounds. When we say galates, then it could be true for the aromatic counterpart, but for the last compound, which is a alcohol, is not true for the Galates. As far as the methodology is concerned, certainly I, being the main architect of methodology of these two compounds, that is Galates and corresponding alcoholic compounds. So first what we did, we took the gallic acid and it was converted into trimethoxy gallic acid. Then gallic acid was converted to acid chloride, or it was kept as such. Once we converted into acid chloride, then it is easy to convert the acid chloride into the corresponding gallate, that is trimethyl gallate. So this consists of first methodology. The second methodology, after a lot of trial with acid chloride, we had some problem with aliphatic counterpart. Therefore, what we decided that trimethoxy galates can be directly converted into the trimethoxy galates. Correspondingly, what we did, we took the acid and converted into the potassium salts, and then the ester was made. Now here are some of the list of the galates which we prepared using the naturally occurring phenols. If we have a diphenol, it can be converted into a monophenol ester if you see the compound number 4A. 4B, in which one of the group was hydroxy, other was methoxy. It was again converted into the corresponding galate. Now, the galates were converted, the galates were directly used. I would like to first give some more examples as far as the 4A, 4B, 4C, 4D, 4E are concerned. We studied them that the ester has formed by IR spectroscopy, because IR is a very good instrument in terms of the functionality in organic compounds or drugs. One can determine the functionality with the help of IR spectrum. And in all these compounds, starting from 4A, 4B, 4C, 4D, and 4E, we got the ester group that was at 1734, 1730, 1727, 1731, and 1737 again for the ester group. Similarly, other spectroscopies were also used. For example, C13 NMR spectrum, mass spectrum. Certainly, the proton NMR spectrum were also recorded for these esters. Once we record the C13 NMR spectrum, the carbonyl carbon appeared, let us say, at 165, 164, 164, and 163 respectively for 4A, 4B, 4C, 4D, and 4E. After the determination of the carbonyl carbon, we went to the mass spectral analysis. Here, I would like to take a minute that ESI was recorded. ESI stands for electrospray ionization. That is a technique by which we could do the ionization much more effectively than the simple ionization of a molecule. And in all these ESI-MS, M+1 was obtained if we do not use the corresponding sodium and potassium ions. In case we use, we can get both M+1 and M+ sodium or potassium peak. The other galates were also prepared. For example, we prepared the galate from the aldehyde. If you see the compound number 4F, certainly one of the hydroxyl group was protected as OCH3. And the IR spectrum suggested that these compounds were formed by the frequencies at 1730, 1731, 1733, 1754 respectively for compounds 4F, 4G, 4H, and 4I. The last compound, if you see, is an alcohol. It is not basically the natural, it is not basically the natural phenol. Rather, it is a natural alcohol. Now, in all the compounds, we could notice the presence of carbonyl carbon as well, except in case where the compounds which we could not see the carbonyl carbon, we discarded those compounds. That means the ester formation did not occur. As far as the mass spectrum is concerned, certainly ESI-MS was recorded, and you will notice that the corresponding M plus sodium peak were observed in the first 3 galates, 4F, 4G, 4H, while it was not observed at in the last compound, that is 4I, where we recorded only the ESI and not having the sodium ion concern. Then we did some evaluation of physico-chemical properties. In physico-chemical properties, we noticed that partition coefficient, solubility, UV spectrum, correspondingly, not only the wavelength, we could determine the extinction coefficient. Extinction coefficient of all the galates gave us the idea that the probability, or let us say the capacity or capability of the esters to undergo the n-pi star excitation. So therefore, the extinction coefficient was very important process. The extinction coefficient was very important data to give us the probability of transition from N to pi star, which we were looking for. After that, we determined the pH value. Certainly, for the standard, we did not, we were not able to find out the pH. And what we could notice that the pH at 7.4 and pH at 1.2, we could determine the physico-chemical properties like K, T 1/2 in terms of minutes and hours. We converted everything into history, and we got the data which has been shown in this table too.
Good morning, sir.
I would like to tell a little bit of the lipophilicity. Which one is water? The water being more denser than an octanol, it was at the lower layer. It was in the lower layer. So we determined the log P, and we could come out of the coefficient log, let us say the log C concentration in octanol and concentration in water, which has been given in the form of the table as well. Now, sometimes the presence of CHO at the para position of the benzene ring may be doubtful, okay? Or we might say that the CHO might change the position of the ester group. This doubt was certainly in the mind of our research worker. So I explained that the ester will give a peak, but because of the presence of CHO at the para position, I can show it by resonance that resonance is stabilizing the O positive next to carbonyl group. Therefore, in compounds, the ester group was not a usual one. Okay, next table 3 shows the anti-inflammatory data and test compound and their physical mixtures in the drug, whatever we use as a standard, induced. Yeah, so whatever here I wanted to talk about, or edema. Here I would like to say that Professor Renu Chadda and Professor Manandar Karan, they help us a lot in choosing the animals, choosing the rats, and then carrying out the testing of these drugs 4A, 4B, 4C, 4D, 4E, F, E, H, I. And we could find out that out of these drugs, some of the drugs were very important. And that's why we decided that we should really test those drugs not once, twice. Okay. So we again got the animals and we could test these drugs which were really potential for us as anti-inflammatory drugs in comparison to test drugs. You will notice that the indomethacin was, in case its dose was 12, our dose of milligram per kg of the rat will certainly was more than that of the standard drugs, TMGA or indomethacin, 12 or 20. The dose of the drugs were more, 28, 30, 33, 32. So that way we can say that the doses were more. Now, I would like to extend the table 3 little bit for the anti-inflammatory activity of physical mixtures with that of the TMGA. Now, you will notice that TMGA, which we have taken, it was mixed with the compounds like 1A, 1B, 1C, 1D, 1E, 1F, 1G, 1H, and 1I. After the mixing of these doses with the standard drug, what we could find out in the animals that only 2 or 3 compounds were active, others were not. And therefore, I would like to say that ulcerogenic activity and anti-ulcer activity, as I have shown in the initial 2 slides, that the ulcer was a important disease which occurred in the stomach. So our compounds like 4A, 4B, 4G, 4H, every compound was tested in terms of dose, ulcer index for the control, as well as for indomethacin were determined. And we could find out that some of the compounds were really active and other compounds were not active. So if you see the table number 4, you will find out that we could say significant as compound to control group, significant as compound to control group, and having different P. Okay. So that way you can guess that some of the compounds were active and all were not really active. Now, I would like to tell a little bit about the binding, how the drug binds with the receptor. Generally, a basic chemist takes the help of van der Waals, dipole-dipole, pi-pi stacking, hydrogen bonding, salt formation. Though one of our counterpart from the pharmacy department had the doubt whether the binding we can pinpoint in terms of these 5 factors. But as a basic chemist, I know that these 5 parameters must be there for the binding. Whether the drug binds with the receptor in terms of van der Waals interactions, dipole-dipole interactions, pi-pi stacking, hydrogen bonding, or salt formation. As a matter of fact, the salt formation is not new. I added this in terms of whether the charge is being completely transferred to the receptor or not. So here I would like to give a little bit of the idea how the dipole-dipole interaction helped in understanding the binding of the drug with the receptor. What we say about the dipole-dipole interaction, we say that it is the electron density. We do not talk about the movement of electron. As a matter of fact, the movement of electron cannot be predicted because the movement of electron can be converted into electron density in terms of the, like we put the positive charge on carbon and we put the negative charge on oxygen. So as a matter of fact, this reflects the electron density. It does not reflect the cloud of electron as such. So what we say? We say that because of this fact, the electron density is most important than that of the electron movement itself. So we should talk about the electron distribution in the molecule, and this electron distribution is responsible for the absorption or for the binding of drug with the receptor. As far as the hydrogen bond is concerned, certainly some of the drugs, they do behave as hydrogen donor or hydrogen acceptor. But here I have shown the hydrogen bond in terms of HF, in terms of carbonyl, and in terms of amine. So all HF, carbonyl, and amine, they are making the hydrogen bond with itself. Let us say HF or carbonyl with alcohol or amine with alcohol. The examples I have given here. As far as the hydrogen bonds are concerned, nowadays the concept of electron density has come. And the electron density can be calculated by some programs. It is not possible to see the electron density just by writing the structure formula of the drug. How can we see the electron density? Let us say just the electron density in water molecule. Oxygen has the electron density which is negative, delta negative, and H has the delta positive. Therefore, hydrogen bond between water molecules occur. Similarly, the hydrogen bond can be formulated to occur between the amines. And this sort of the hydrogen bonding has already been shown in DNA. In nature, DNA stores, transfers, reproduces genetic information. So this first sentence is responsible when we talk about that how DNA stores, how it transfers, how it reproduces the genetic information from parent to the kids. So all three processes occur because of the double helix structure, and that double helix structure is maintained by hydrogen bonds only. Okay? Otherwise, there is no two complementary linear strains which can hold the molecules. Now, when we talk about the hydrophobic effects, hydrogen bond and pi-pi stacking interaction also occurs. Certainly, other than hydrogen bond, hydrophobic and pi-pi stacking, which other students and some of the teachers, they cannot understand the pi-pi stacking very well. So what I would like to say that they must not write a aromatic ring over it. They should first see the electron density. Electrons density is more in between the aromatic ring, while it is less at the periphery. Therefore, the aromatic ring can bind T-shape or it can bind with slip stacked. Therefore, the binding of the drug with receptor occurs differently. Certainly, fluorine was not present in our either in alcohol, phenol, or the Galates. I would like to tell a little bit about the Lewis acid-base interaction. Certainly, in some cases, this occurs. I have shown here the interaction between boron trifluoride and dimethyl ether, which is a very well-known example in the laboratory. That how did boron trifluoride binds with dimethyl ether? As a matter of fact, boron trifluoride is a gas, but it is kept in the solution when acid-base complex through the Lewis acid-base interaction occurs. Okay. Now I would like to come to the programs which we have used, a Schrodinger program made in 2011, and it was the reference for that program is of Journal of Medicinal Chemistry. If one can summarize the docking studies, I would like to give the example or to the layman that it is simple as hand in the glove. When we say hand in the glove, what is the hand and what is the glove? Hand is basically the drug and glove is basically the receptor. Therefore, molecular docking is not difficult to understand by keeping the hand in glove idea in mind. So whatever study we did with our compounds, we did the theoretical study as well as the practical study. Practical or experimental studies I have already shown. And when we did the molecular docking studies, we put the structure formula on the computer screen and got this program with the receptor. And we could see that some of the drugs were really showing the effect that they can bind very effectively. But the other drugs, they could not bind effectively. And for these, we collected the data from the molecular docking program. And as a matter of fact, you will see that we could we could see the binding of all the drugs with the receptors, and we could calculate some of the data given in table 6. And some of the data, I would not like to talk about the molecular weight, the other things, but I would like to talk about the donor acceptor, Q log, whatever QP log P0, etc. over W. So these are the binding parameters by which we can say that molecular properties of test compounds starting from 4A to 4I can be very well understood, keeping the standards in mind. Indomethacin, Rofac, Cosib, diclofenac, sodium, etc. These drugs we bought from the market. As a matter of fact, our two professors from the pharmacy school of Punjab University helped a lot in calculations, particularly calculating the molecular properties. After this, I would like to conclude my talk by saying that cycloalkyl or aryl 3,4,5 trimethyl showed promising anti-inflammatory activity. All galates, they shows some activity. But out of these compounds, only 4A, 4B, 4G, and 4H, only 4 compounds emerged as the most active from the data as well as our experimental studies. The results of the ulcerogenic studies and biochemical evaluations suggest that these compounds are gastric safe. That means they can be used as the drugs. Docking studies are in good agreement. Certainly, it is a theoretical data. I would say that the docking studies helped us in understanding the experimental results. But I don't rely a lot on docking studies, to be very frank. But because of the fact that the docking studies gave us the data, certainly for 4A, 4B, 4G, and 4H were good enough, but not a lot of good. This investigation suggests that the galates are potent, new gastric safe anti-inflammatory agents. At the end, I would like to acknowledge my appreciation for the research worker, for example, Mamta, Pran Kishore, and Professor Renu Chadha and Professor Maninder Karan. As a matter of fact, the student Mamta Sajdev Dhingra did a lot of work. And she was the last student of Professor P.D. Sharma, who died meanwhile we were doing the preparation of these compounds. We did not do the experimental and docking studies at that time. Okay, after this, you know that the funding agencies like UGC and CSIR helped us a lot for financial support. Therefore, I would like to thank both UGC and CSIR. And the research scholar got some of the meritorious student scheme award for the senior research fellowships. So I would like to acknowledge the UGC and CSIR for this award. Also, I would like to acknowledge the support of Punjab University in terms of the data we collected for NMR and other spectral data. I would like to give a special tribute to late Professor Pritam Dev Sharma, one of the investigators at the beginning. Last but not least, I would like to thank everybody for the patient hearing. In case anybody has a question, Madam Shalini will certainly ask the audience to ask the question.
Thank you, Professor Tej Vir sir, for sharing an informative talk on Galates. Now, may I please request our panelist, Honorable Professor TR Bhardwaj sir, and Professor V.K. Kapoor sir to please start the Q&A session. Over to Bhardwaj sir.
Thank you, Shalini. It was very good approach with respect to the side effect which has been again seen in the case of the non-steroidal anti-inflammatory agents. Most of these non-steroidal anti-inflammatory agents, they have free carboxylic acid and results into the hyperacidity, ultimately leading to the ulcers, peptic ulcers. So by preparing Galates of the gallic acid and others also, Professor Tej Vir Singh and his co-workers have done very significant work in the area of non-steroidal anti-inflammatory agents. So I must congratulate Dr. Tej Vir and his team, specifically Dr. P.D. Sharma, co-worker, and other students also. What has been my observation with respect to the non-steroidal and anti-inflammatory agents, specifically having phenolic OH? In such cases, some liver toxicity has been reported or observed. So, with respect to the liver toxicity of these molecules, was that also studied or it's still pending? The liver toxicity was not detected in the case of the animal studies, or it was not done.
As a matter of fact, we have not studied at all
I see.
the acidity in liver, etc.
But I think it's a very good molecule. It should be, again, it has already been published. Otherwise, I would have suggested that it should have been patented and commercialized.
Yeah, so your suggestion is well taken. Yeah,
very good. Yeah, over to Madam Shalini.
Thank you sir. Over to Kapoor sir.
Thank you, Shalini. The work presented by Professor Tej Vir Singh is an example, excellent example, how natural products, particularly those which are present in abundance in nature, could be utilized as medicinal agent. I mean, Galates, gallic acid, they are present in abundance in almost all the plants. And as such, they don't exhibit any biological activity. But the work carried out by his team is a novel example that how biologically inactive compounds can be converted into useful compounds having some activity, pharmacological activity. So it is indeed a very novel work which has been presented by Professor Tej Vir Singh and his team to be congratulated for this. My only query was with regard to the standard used in comparing the activities of the prepared molecules. I mean, they have used indomethacin. What has been the basis? Because it has anti-inflammatory, non-steroidal anti-inflammatory agents. They have wide categories of compounds. Why particularly an indole molecule was chosen?
Yeah, let me answer this question in terms of our funding. we did not have the funds to buy the really expensive other drugs which can be used as standard.
Okay, okay. That's understood. Nevertheless, the work carried out is very significant, and I think more esters with different functionalities and peripheral groups could be prepared and their biological activity be shows that a lot of work can be carried out in this direction in future. I once again compliment Professor Tej Vir Singh for his presentation. Thank you.
I thank you, Professor Bhardwaj and Professor Kapoor, for the suggestions.
Thank you.
Professor Mishra?
Thank you, Kapoor sir.
Thank you.
Participants, please raise your hands to ask question. Shivani ma'am, please let Ekta Singh in.
Am I audible?
Yes,
Yeah.
yes, you are audible.
Good morning, everyone. Good morning to the panelist and the speaker. I would like to congratulate the speaker, Dr. Tej Vir Singh sir, for his wonderful presentation and insightful research. Sir, my question is regarding interaction. Though you mentioned that dogging results are not, you do not appreciate that much, but you mentioned about the interactions. Among those interactions, which is the most important? Because nowadays docking is a fashion. Everybody wants to have in silico result that predictable binding interactions are happening. And if the distance of all the interactions considered to be same, then which interaction is the most significant? Thank you, sir.
Yeah, I would like to answer it this way, that finally the experimental results are the supreme. I believe that whatever the experimental results were, they were not exactly the same as we did the theory.
Tejveer sir, you are not audible. Hello? 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 There is some technical issue with Tejveer sir.
With Dr. Tej Vir?
Yeah, now I am audible. Yes.
Yes.
So
Yes, sir, you are audible now.
let me answer the second part, that the receptor is so big, whatever calculations you do, you may not be very sure. But certainly the experimental findings are the best. Whatever the way you do, you please rely on experimental findings. Don't rely a lot on docking studies. Certainly many people do the docking studies. As far as the second part of your question is that which out of 5-6 studies which we did, I would say it is the hydrogen bonding, which was almost there with the experimental finding. Now, the question is that hydrogen bond not only depends on length, not only depends on bond length, it depends on bond angle too, bond energy too. We never determined the bond angle, bond energy, or bond length because the receptor is so big, therefore we were unable to find out the real bond length, real bond angle, real bond energy. So hydrogen bond was certainly successful, but the problem was to see its strength, whether we can rely a lot on it or not. Therefore, we did all these studies, but the data, whatever we got from the program, that is appreciable. It is not exactly as that of the experimental finding. I think I answered your question.
Thank you, sir. Shivani ma'am, is there any more question?
No, ma'am.
Okay. Thank you, Professor Tej Vir sir. Thank you, Bhardwaj sir. Thank you, Kapoor sir.
Thank you.
Participants, now we are going to welcome our next keynote speaker, Professor Dr. TR Bhardwaj sir, Advisor to Chancellor, Baddi University of Emerging Sciences and Technology, Baddi. He is former Chairman, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Dean, Faculty of Pharmaceutical Sciences, Punjab University, Chandigarh. International Research Fellow, SRI International, formerly Stanford Research Institute, California, USA. Former Director of Sophisticated Analytical Instrumentation Facility and former Director, PG Studies, ISF College of Pharmacy, Moga. He has guided more than 70 pharmacy students and more than 14 PhD students and published more than 120 international research papers and the most 16 patents including 6 US patents. He is Executive Committee member AICTE, National Advisory Committee GPAT, UGC nominated member for Special Assistant Program. May I please request Professor Dr. TR Bhardwaj sir to please share your presentation. Over to Bhardwaj sir.
Thank you, Shalini. The slide is visible or? Hello.
Sir, your voice is audible, but slide is not visible.
Okay.
There is some network issue.
Yeah, we are. Is it visible? Hello, is it visible now?
Yeah, yes.
Yes, it is visible, sir.
May I start, sir?
Yes, sir. Please.
Okay. Good day, everybody. I'll be talking today on the topic Emergence of India as a global health provider. As we all know that over the decades, on account of unstinted efforts of our fellow pharmacists and professionals, India has earned the present status and positions globally, and we are among the 3 top countries who are again pioneer in the area of pharmaceuticals specifically. However, with respect to the health provider, India has never been behind. Since ages, India has always been contributing in the global health. And we had again attained better positions or better contributions during COVID-19, 2019 pandemic also. At that time, when the oximeters, masks, and number of other medicines, specifically hydroxychloroquine, which was again at that time considered to be one of the drug in the prevention, India was able to synthesize and give it to the more than 70 or 75 countries throughout the world, provided that. So that capability potential of India is having. Taking into consideration these capability and potentials, in June 2020, US Ambassador Sandeep Chakravarti, Consulate General of India at New York, in collaboration with Global India Trade and Cultural Council of USA, Mr. Harinder Singh Panesar, they organized Augmenting US-India Healthcare Relations under COVID Challenges. And we were again lucky, we were rather happy to participate, fortunate to participate in that webinar. And during that webinar, one of the speaker, Mr. T. Srinivas, he was from Bharat Biotech. He said and gave the present statement at that time that we are working on vaccine for COVID-19, and we have seen the result there afterwards, that Covaxin was outcome of it, and thereafterwards, of course, the Oxford and AstraZeneca, their vaccine was further taken by Serum from Pune. So those kind of contributions have also been a matter of pride for all of us. Although I will be mainly talking about the drugs and pharmaceuticals because these are the pharmaceuticals which are again their contributions as compared to the other approaches with respect to the healthcare have been very significant. If you look at our prior or the positions before 2005, when the product patent was again started or it was again implemented by the regulatories in India also. Prior to that, we used to have process patenting and taking care and opportunities from the patent process patenting. Dr. Reddy, NG Reddy, and Ramarao from Hyderabad, they came up with some alternative processes for the drugs which were again generic or whose patents expired. That gave an impetus to the pharma company, because as we all know, to invent new chemicals and take it to the clinical phase 3 level or clinical applications, it requires huge amount investment. Out of maybe 10,000 molecules, 10 may reach the clinical trial, and you are lucky if even if one of them becomes drug afterwards. And it has also been seen that none of the drug has been reported or come out of educational institutions. In that case also, the University Institute of Pharmaceutical Sciences, Punjab University, we were lucky to have our late Professor Harkishan Singh, who could provide xanthonium iodide, which is again neuromuscular blockers, to the rest of the world. It was again drug which was invented at the University Institute of Pharmaceutical Sciences by Professor Harkishan Singh and his group, and was found to be better than what were available at that time. Panchronium iodide, xanthonium iodide was the shortest acting drug with respect to the neuromuscular blocking activity. So we have never been behind the rest of the researchers globally. If we look at the Indian pharmaceutical evolutions, it has 5 phases. Phase 1, domination of foreign companies. Prior to 1970, if we look at, I think more than 80% of pharmaceutical companies, they were housed in Maharashtra and Bombay in particular. It may be X, it may be Ciba-Geigy, it may be Glaxo, all these were there only. And their research units were also at Bombay. And they were again taking care of, or they were the ruling the Indian pharma industry. However, around 1980, when the phase 2 government control was there initially, and of course, around 84 or so, when the process patenting was again used extensively by not only the Indian pharma companies, they could enter the global market. They were preparing all those raw materials, APIs, or the bulk drugs at much cheaper cost, and they were supplying to the rest of the world. And thus, it was made accessible to the poor population also. You see. Then came phase 3, development phase, and then phase 4, growth phase, when the multinationals were almost, you see, they have their branch, or they have their company in India also. They started manufacturing, and phase 5, innovation and research, which after maybe 2010 or so. Because of, you see, we have the manpower, we have the raw material. Therefore, because of these 2 things and other facilities also, support from the government of India also, the cost of the drug was minimum or very cheap comparatively. The only competitor of Indian pharma company was and is China, who are having slightly the drugs which are being manufactured of bulk drug and APIs at slightly lesser cost. Therefore, we are importing some of the intermediates, API, from China. And it's a matter of pride for us that these days globally you will find formulations made from India, and that it's again matter of, it's proud to be in these professions, that out of those 1/3 are being manufactured or the formulations are being prepared in our neighborhood at Baddi University of Emerging Science and Technology, School of Pharmacy and Emerging Sciences. In this area, there are almost all companies have their units over here, you see. And another good news is that recently Government of India has again sanctioned, and money has been, I think, given to the Himachal government to start bulk drug park also. Out of 4 bulk drug parks which are again going to be established in India, one is near Una, which is again maybe around 100 kilometers from Baddi. In addition, diagnostics are also coming up at Nalagarh. So BVNIA, that is Baddi, Brotiwala, Nalagarh Industrial Association, or the area, is going to be again boosted for manufacturing, for APIs, for formulations. So all these are going to be again further augmented in the nearest future. So let us take a stock of what are our strengths, weaknesses, what's known as the SWOT analysis. Our strengths are cost effective. We have very strong manufacturing base, availability of high quality skilled workforce, and diverse ecosystem. It's really very encouraging and surprising also to see that the Baddi or this area, there may be not less than 60 or 70 companies, manufacturing units. All these are again approved by FDA, Food Drug Administration of USA, as well as European also. So they are not manufacturing exclusively, no, they are manufacturing exclusively for the rest of the world. Same is true with respect to the Fresenius Kabi also, who are working in the area of, again, anti-cancer injections or the drugs. So most of the companies, that means you can say in 1/5 or 1/6 of the companies, they are making drugs and pharmaceuticals for export only, you see. That means the quality is equally good, cost is minimized, and these are the reasons that you can provide the quality, maintain the standard, and supply it the rest of the world, you can definitely fetch the market. What are our weaknesses? That is less investment in R&D. Governments are again investing miserably to the pharmaceutical research. That's one of the area, and that's the reason that our contributions in the new chemical entities, new drugs, new molecule, which requires huge amount of investment, is minimum, or you can say it's negligible almost. So lack of eco-coordination between industry and academia. Although some of the institutions and multinationals or the national research units, institutions like CDRI or IIIM Jammu and these kind of research units who are working in the area of research for NCE, companies, industries, they want immediate results with minimum investment. So that is again another hurdle. We at Baddi also, we expect that we have very good relationship, we have very good coordination. They are our collaborator also. But as far as R&D is concerned or other these things concerned with respect to research and development, they have either they are not having faith or they don't want to go for it. They want to continue with their routine work, you see. That means they want to complete their quota preparations or the supply. They don't want to improve or they want to enter into the new chemical entities, businesses. And then negligible expenditure on healthcare in the country. In India, healthcare is again given minimum sanctions as compared to the rest of the countries in the world. What are our opportunities? That is increased export potentials. So the profit margin, when the drugs are or the formulations are exported, is very, very high comparatively as compared to the drugs, generic drugs specifically for Indian market. So that's one area which can again provide financial support